Hi everyone, I'm very happy to be today for Enlace Judío with Professor Jonathan Gershoni from Tel Aviv University. He is in the Department of Cell Research and Immunology, and he has been awarded several times Teacher of the Year by the University of Tel Aviv. So I'm super happy to be with you, Professor. Hopefully you can teach me lots of things about biochemistry and immunology.
I'll do my best.
Okay, great. That's enough. And we're going to talk a little bit about Corona, obviously, and a little bit about immunology in Corona and your work. And we want to understand the relevance of your participation in Innovation Day for Tel Aviv University here in Mexico for 2020. So, welcome. And I would like to start by asking you, I saw something you wrote in the profile of your lab about inventive research. I understand what basic science is. Basic science is the answer to basic questions without really wanting to know how to apply them, but just for the pleasure of knowledge. And then we know exactly what applied science is, no? But what is inventive research?
Well, very early in my career, it became evident that whereas I understand the importance and the beauty of doing science for basically trying to understand mechanisms in themselves, I always found myself asking the question, well, what can I do with it? How can I apply it? How can I use my knowledge as a means to promote and improve human health? So this is a question that's been very important for me. Now, on the other hand, at the complete extreme of applied biology, then, you have a lot of very serious questions, let's say in the industry, as to how to improve the cost benefit in production, or maybe increase the shelf life of a product. And I felt, well, that's too applied. And very early as I was working, I realized, not something that I intended to do, but I came up with some ideas. And at the university, they explained to me that actually I'm an inventor. And I hadn't planned on being an inventor, but I'd come up with some new concepts and new technologies. And the university said, we have to protect your invention. And that was the first time I became aware of patents and inventiveness. And I began to understand that the criteria, the definition of a patentable invention is it has to satisfy a number of basic rules. First of all, it has to be useful. So inventive research, because ultimately I said, okay, this is what I'm doing. Inventive research means that it first and foremost has to be useful. Secondly, it has to be novel. Novel means that there is nobody who's done exactly what you are doing in the world. And so it has to be completely novel. And second or thirdly, it has to be what patent law says, unobvious to one with average knowledge of the art. Meaning that if I'm looking with my colleagues and they look at what I'm doing, they're either going to say, oh, that's cute, or they're going to say, I've seen this before. There's prior art. And so what you've done is obvious to anybody who's really familiar. So for a patent, it has to be novel, it has to be useful, it has to be unobvious. Now, in addition to that, in order to gain a patent, you have to be able to describe your invention in terms that will actually teach somebody who's familiar in your field, or what we refer to as an enabling disclosure. And you don't necessarily have to reduce to practice. You don't have to actually do the invention, but that would be a good idea. And so over the years, I've had the opportunity to file for numerous patents and inventions and been awarded patents for my inventions. And so when I was asked at one time, what are you actually doing? What kind of a scientist are you? And I typically say I'm a biologist, but then they say, well, what kind? Are you applied biologist or do you do basic research? And I said, you know, I think the definitions of a patentable science is very wholesome to me and very productive and very efficient because it keeps me focused on trying to do, as I said, novel things of use, sharing that with others to teach them my art and to do things which are not obvious. And I've been blessed because over the years, I've been able to secure quite a few patents in the fields I'm interested in. And so at some point, and maybe it's not by chance, because apparently I have an inventive mind, when asked what kind of science you do, I said, I do inventive research. Now, not only is that interesting in a sense, but it's a field which bridges two disciplines. I'm a college professor. I teach and I do research in the university. We're not an industry. I would like to interact with the industry and share what we've discovered and invented so it can be implemented and used. And so inventive research is that area that bridges between academia and the industry. And it's very important because people have a misconception of the purpose of a patent. They sort of view me as, oh, he wants to make money. Now, don't get me wrong. I wouldn't mind making money, but that's not my objective. However, if you have a good idea, which can actually be useful, it is your moral responsibility, especially if you're using public funds to develop your ideas, to provide the industry with protection so they can invest in your idea, develop a product, and bring it to market. And if you do not provide them with the protection of a patent, as good as your ideas may be, they will never reach the public. Correct. And so patenting is an extremely important tool in order to bridge from academia and bring it to market to people. And so you asked me, what is inventive research? That is inventive research.
It's that transfer.
Exactly.
And so my next question is obvious, which is, which of those patents, I would love to know, which is your proud baby?
Well, if you're familiar with translating patents into products, you know that this is a complex and long process. I've had marginal success. And so very early on, we developed various tools for solid phase immunoassays, and they have been implemented. We came up with a concept which is now being explored years after my patents have expired. So I don't perceive to see, but I've taught the field. And we have come up with the concept of using receptors for viruses as potential therapeutics. Okay. And it's interesting, I came up with the idea that a mimic receptor could possibly intercept a virus and prevent infection. And I had a friend named Rodolfo Linas. He's a professor in New York. And he says, you know, this is a really cool idea. You have to name it. And I said, what do you mean, name it? He said, well, if you call it a mimic receptor, nobody will remember. Give it a name. Because if it has any importance, people are going to develop it. And then years later, somebody's going to say, I wonder who was the first person who came up with this concept. And if it has a name and you named it, there's some chance that maybe people will remember that it was you. So being Israeli, I said I have to give it a name in Hebrew.
Okay.
And I'm thinking in terms of a molecule, a mimic receptor, that the function is to suppress a viral infection as a drug. And the word for suppression in Hebrew is decoy. So I called them molecular decoys. It is a molecule that intercepts a virus. It is a decoy, but it stems from the Hebrew word suppression, decoy. And since then, the whole concept of using molecular decoys and mimic receptors has stemmed from my original work way back in 1983. So it's been quite a few years.
Yes.
And now it is gradually becoming something that people recognize. And the last patent I'll describe, because it leads us into Corona, is that 15 years ago, I was giving lectures in the United States, in Harvard, and a colleague of mine who was listening to my lectures, I was describing methods in understanding the interaction between HIV envelope protein, GP120, and how it interacts with its receptor, CD4. And we have developed some computational algorithms which allow us to better understand these dynamics. And he said, you know, maybe we can collaborate. Would you come to my office and let's discuss the virus that I'm studying? And I said, sure, why not? And we met. His name is Professor Wayne Marasco from Harvard, from the medical school. And Wayne introduced me to a virus that he was studying in 2004, and it was SARS coronavirus. So this was the first coronavirus, and I started working on the mechanisms of how SARS coronavirus 1 interacts with its receptor ACE2.
Okay, so now that you started talking about corona,
yes.
What I want you to explain to me and to the audience is, as HIV, that is one of the viruses you've been studying all your life. You've been in the front of the task force in Israel for AIDS. Mm-hmm. And so studying HIV and the mechanism with which HIV introduces into the cells, which is also in a receptor. Exactly. Okay. So how corona, which is so similar, the SARS-CoV-1 and SARS-CoV-2 that is affecting today's pandemic, that is causing today's pandemic, how all this that you learned there apply to SARS-CoV-1, and then suddenly this SARS-CoV-2 appears and you're ready. You have the tools already to embrace the challenge.
Exactly. So HIV and AIDS is a very different beast. It's a virus that is constantly and dynamically changing. And because it's mutating very, very much and constantly changing, my hypothesis was that there is a surface on HIV that is not allowed to change. It must be conserved. It must be maintained and very stable. And that is the precise area of HIV that interacts with its counterpart in the human cell, the protein CD4. Because CD4 is the docking, it's the receptor. And so the virus can't afford to change the docking onto CD4 because CD4 is stable. And so if we can learn how to target the receptor binding site of HIV, that might give us some indication as to how to either devise better therapeutics or potentially a vaccine. And this is what we were working on.
Correct.
Once we were introduced to coronavirus, it's the same question. It's precisely the same. The coronavirus has a very large protein, 1,200 amino acids, the spike. But if you understand the details and the mechanisms and the structure of the spike, you will discover a relatively small feature of maybe 50 amino acids. And this is precisely, exactly, the receptor binding surface that interacts with the very stable,
AC2,
unchanging ACE2. And so when Wayne asked me, maybe we can apply what we've learned on HIV to SARS corona, this is where we began to collaborate. Now, we worked on this for a few years on SARS corona 1. And by the time we actually learned methods in being able to produce and to extract out the receptor binding motif, what we call the RBM, from the spike of SARS corona and reconstitute it, produce it in a fashion that looks like the native receptor binding motif, but out of context. By the time we did this, nobody could care less. And the reason is that fortunately, SARS corona 1 petered out and disappeared. But, and I say this with care, fortunately for me, there was an emergence of MERS coronavirus in 2012. So this is another coronavirus. Unfortunately, it has a greater fatality rate of about 30%. Fortunately, it's not terribly infectious, but the moment we realized that there is a similar virus in the same family of beta coronaviruses, we realized that we might be able to implement what we learned from SARS into this new virus in 2012, and that's what we did. So we developed all of the tools initially for SARS and then implementing for MERS. And in 2015, it dawned on me that actually I have now an in-depth understanding of coronaviruses, their receptor binding elements, and how they interact with their receptors. And so we filed for patent. And we filed for patent in 2015. And the title of the patent is Vaccines for Coronaviruses. And this is in 2015. And we were in the process of writing up our data on the MERS, because we published for SARS. So we were in the process, and then, as we all know, in December of 2019 in Wuhan, there was the emergence of a new threat. And so we were precisely ready because we had worked 15 years in understanding the details and the mechanisms of how coronaviruses bind and interact with their receptors and how to translate that into a new modality of a vaccine, which we're working on today. And we're making progress. It takes time. These are very complicated type of experiments, but I'm confident that because of the similarity of SARS coronavirus 2 and with SARS coronavirus 1, we will certainly be able to reconstitute the RBM, the receptor binding motif. And this should be, in my mind, and hopefully, a very important component in combating SARS and developing a vaccine modality. And so this is what we're doing today.
Wonderful, wonderful. It's incredible how you were building the blocks without knowing exactly the future. And when the future came, you were ready. You were ready
Yes.
to work on this. And so
this is very, very bizarre.
It's serendipity.
However, it falls very much within my general philosophy of inventive research, of trying to do things which have utility, and working for 15 years on coronavirus, possibly 30 some years on HIV, as well as HCV and flu and dengue. So looking at the dynamics of how viruses interact with their receptors, we were very prepared to now undertake the challenge of SARS coronavirus 2.
Okay, and at what stage now, because what happened to you, which is serendipity, really.
Absolutely.
What happened to you was it's not what happened to other labs. Most of the labs right now which are working on corona were not working on corona prior to the pandemic. They had to stop everything, stop their research, and focus on this new challenge. And on the contrary, you were already on tracks. You had your inertia going, you had your momentum, and so you only had to understand how to make your research useful for this emergency. And so the question is, what is exactly the property of the vaccine you're building compared to the other forms of vaccines that are out there, that are already in the races, that are already, I'm talking about the Oxford and the CanSino and the Moderna and now Johnson & Johnson. So what's different about yours?
Okay, so first of all, whereas everybody assumes there's a competition here, there's a race, who will be first, it's not that important who will be first. What is important is that we need a solution and a vaccine.
Yes, because first doesn't mean successful or safe or effective.
Okay, now we are being constantly bombarded by press releases that Moderna has made another step, and AstraZeneca and Oxford have made another step, as well as Pfizer and all the rest. For the general public, we shouldn't care less. The only thing that the public is really interested in is, when can I go to my health plan, roll up my sleeve, get a shot, and know that I and my family are safe and we can get on with life and enjoy the next vacation. That is what we're looking at. And so all of the reports that there are encouraging indications in a phase 1, phase 2 are all technical. And the question is, when will we actually have the vaccine that I just described, something that will protect our families? And I don't believe that we're going to see such a vaccine which will be available to the general public in Mexico or in Israel much before a year from today. And if that's the case, that will be wonderful. Which means basically that for at least the coming next year, we have to learn how to live with corona.
Okay.
Now, how do I distinguish myself from all of the other modalities? Surprisingly to me, I would say that most of all of the vaccines, and it could be Johnson & Johnson, AstraZeneca, Putin with adeno 5 and adeno 26, and Moderna, all of them are all trying to present to the immune system the spike protein, the envelope protein of the virus, in various forms and shapes. You have some vaccines which are like software vaccines. They're gene transferring. So Moderna is giving you RNA, which codes for the spike.
Yes.
Johnson & Johnson is using adeno, which is a non-replicating virus vector, so as to deliver the gene for the spike. And so these are vaccine modalities that are trying to provide the vaccinee with genetic information to produce the viral antigen on his own. So it's like software. It's like using a disk on key.
Okay, but the Oxford one is using the spike, right?
The Oxford one is using a chimpanzee
adenovirus.
Oxford adenovirus, ChAdOx,
yes,
which packages the gene for the spike.
Okay.
And when you inject the ChAdOx with the gene inside, that gene is expressed and therefore generates and stimulates an antibody response to the protein that the vaccinee expresses.
Correct.
Now, a hardware vaccine would be a vaccine where you actually inject the antigen.
Yes.
So you can make a subunit vaccine, let's say Novavax, has the spike protein being produced in insect cells, and that protein is being directly injected into people in the same fashion as the hepatitis B surface antigen protein is being injected into babies. So this is a well-established, what we refer to as a subunit vaccine modality. CanSino and 5 other companies in China are using a different approach. They're saying, let's grow up the virus and let's inactivate it with beta-propiolactone. And so now you have an inactivated virus, exactly as did Salk with polio.
That was,
so
I was
we have
going
an
to tell you.
inactivated
That's the vaccine. That's how
So I'm saying these are the classical virus vaccines.
Yes.
You have subunit vaccines and you have inactivated virus. And so unfortunately, in my mind, everybody's being enchanted with the new technologies, gene transfer. Everybody's saying, well, Moderna's coming up with something spectacular. However, we have to be a bit more realistic. We have to realize that there is not one single RNA vaccine on the market today.
Correct.
So it's a totally new technology. There is not one adenovirus vaccine today in the market for human use. And so whereas there is an enchantment, and it's exciting to think of cutting edge technologies, if you ask me, I would prefer to see a traditional vaccine being developed. A subunit vaccine with the spike protein on the one hand, and on the other hand, possibly an inactivated virus. Now, all of the vaccines, inactivated virus, subunit, RNA, adeno, they're all intended to present the intact spike. And in the world of coronaviruses, sometimes antibodies directed to areas of the virus which do not actually prevent infection, which are not effective in knocking out the virus, this excess antibody is not only not beneficial, it could be detrimental. There's a phenomenon called antibody-dependent enhancement, and it's very common, for example, in dengue, and we're studying dengue as well. And so our solution to these situations argue that what you really want to do is you would like to vaccinate a person with the most vulnerable Achilles heel, if you will, of the virus. And that is the receptor binding motif. And so whereas everybody's developing vaccines using the spike, we have a patented technology. So we're the only lab in the world that has the right to say, let's reconstitute, extract out precisely, precision, just the binding surface of the virus and use that to train the immune system. Now, as far as the competition is concerned, it is clear that we are a small lab in Israel, and we are not able to compete with Merck, Pfizer, Johnson & Johnson, Sanofi, GlaxoSmithKline, and even Moderna. We're a small lab. But first of all, we have the patent. And so if any of the companies would like to implement a reconstituted epitope, that would be fine. However, my perception is that it really doesn't matter which vaccine comes to market. Whatever the vaccine may be, very common is the fact that effective protective vaccination entails a primary inoculation and then subsequent boosts. And in my mind, we will have the ultimate boost, because whatever the vaccine was, it will stimulate a spectrum of antibodies towards various epitopes which are irrelevant in the spike, and hopefully a proportion of the response will be to precisely the receptor binding motif. And then it will be advantageous to come in with a boost, which will then specifically stimulate the immune response to precisely where you want it. And so the question is can other people do the same? If so, I don't know how. Can we do it? That is our hope. We are hoping that our modality is definitely, I mean, we have a US patent, so it satisfies useful, novel, unobvious, enabling disclosure and reduction to practice. And hopefully we will get it done in the near coming months.
And well, I think that's a marvelous approach, which is a very precise way to working, like as it's clean and it's elegant. But I would love to know if there is any other vaccine in the market that we use already approved with this approach in a receptor binding material. Okay.
No.
Okay.
So in reality,
okay.
So we're in a funny
this
situation.
Is novel. This is also a new technology.
Exactly.
Okay, no, that's great. I think it makes sense.
So the question is there a proven, what I refer to as epitope-based vaccine to the precise receptor binding site in the market and being used? And the answer is no.
Okay.
We've been working for many years in trying to develop this as a concept. And then here again, it is fortuitous that all of a sudden, we believe that our approach is not only, as you said, very precise and elegant, thank you very much, but in addition, it's very, very much to the point of this current horrific pandemic. And so we are encouraged that the stars have the horizon and Saturn way out there. The stars have been such that exactly as we are developing this new approach, which we believe should be very effective, all of a sudden also has an enormous application. So I feel that I've been working on a play for 40 years. I've been writing a script, and all of a sudden I've been given a stage.
So you're not doing inventive research anymore, you're doing applied science.
Well, maybe. I'm still working on the invention.
Well, the transition. So sure. I'd love to know, with your great understanding of the immune system and the response of viral infections in the immune system, if there is any idea, or if you have any idea how to break down the wide range of responses that we're getting with corona. You see asymptomatics, you see people with a wide range of different symptoms, even the long haulers, which have symptoms after 3 months of the infection. And with there is a group that described already 100 different symptoms, from hair loss to peripheral neuropathies. So what's happening with the immune system? What is this corona doing so differently in us? Because I understand, obviously, it's the same virus. What is different is our response. So how is the corona tricking or how do you say it, like triggering so different responses? It's the same virus. How is it capable to do so?
So here I can come to the typical answer is, I have no idea. Now, in a more serious vein, I first of all have to point out I'm not a real doctor. I'm not a physician. I'm just a scientist.
But you understand immunology.
Yes. So I understand some aspects of the virus, immune system, human body, pathogenesis system. I have no real insights as to why this virus is so nasty and stimulating such a vast response. There is one aspect of everything you've said which I find very curious and possibly relevant to what I know how to do. And that is, it appears that this virus, as is in the case of other coronaviruses, it appears that it doesn't really establish a robust and durable immunological memory. There have been numerous reports about people who have been infected, have raised an immune response, and as people are monitoring the level of their antibodies, at least in some people, they're watching as the level constantly drops. And there doesn't seem to be a persistence and a durability to the response. Now, I don't know if that's truly the case, but this might be a unique situation. It could be, if I was a coronavirus, it could be that it's to the advantage of the virus to actually infect an individual, stimulate an immune response which is sufficient to clear the virus, but not aggravating to the extent as to establish a very robust and effective memory, which means that next year I can infect you again. And so if the virus has evolved to stimulate neutralizing antibodies, but not to the degree of depositing B cell memory cells to the extent that I will have a very robust active recall next time I encounter the virus and will be fully immune, then this could be a different type of behavior of virus. Now, why am I encouraged by this? Obviously, I would prefer to have a robust immune response. But I'm saying that this could be a unique situation where the naturally gained immunity through the disease is transient. It's not long-lasting. It's short-lived, because this could be the nature of the virus. And therefore, natural immunity may not necessarily be the best answer. And quite the contrary. Maybe vaccines, commercial vaccines that we're making, we have to be able to stimulate the immune system and repeatedly stimulate with boost and possibly adjuvant and establish a robust and durable immune response that the natural disease has evolved not to do. And so here we have a situation where it could be that the protection gained by vaccination may have a preferred durability as opposed to the naturally developed immunity through disease.
Now,
and so I'm saying this could be at least one advantage of vaccination.
correct. But then will vaccination promote only humoral response or also cellular? Because natural infections promotes also T cells and this robust response, which also is protective. It's not only antibodies, it's also cells.
So generally speaking, you're absolutely correct. And if we were to use exclusively only the vaccine that I am providing, then that would only be the humoral antibody response. We know, for example, the humoral antibody response seems to be sufficiently effective for polio, because inactivated poliovirus, and for that matter, inactivated flu virus, they do not stimulate your T cell immunity, and certainly not CTL. However, I think you're right, and we still don't know if, in addition to the B cell humoral response, do we need a robust T cell response? And this is where possibly the use of the replicating viruses such as VSV, Merck is developing the VSV corona.
What is
VSV?
VSV?
Is vesicular stomatitis virus.
Okay.
And Merck is also using a measles virus vector as a replicating virus. adenoviruses also. So anything that is expressing the proteins by our own cells have the ability to then present the exogenous protein in the context of MHC class 1 and class 2. So I don't want to get too technical.
No.
But,
yes.
but this is certainly a point well taken. And the epitope-based vaccine that we are promoting does not address T-cell immunity.
Okay, so I think it's a very interesting talk, what we've been discussing, because you are the man who, without knowing, has been preparing for this show, and now it's showtime, and you're there with everything, and we're gonna see you soon because you will be part of the Innovation Day 2020, and you're gonna talk on September 30, and your talk is called How Close is a Corona Vaccine? So would you like to invite everyone to be there on September 30 to listen to you? Just
please, hola Mexico.
Hola.
Please come, and I look forward to seeing you. Hasta luego.
Good. And I think this talk is a good advancement on what we'll hear and what we'll learn in a couple of days. I really want to thank you for your time. I know it's late now in Israel, but thank you for your time and for your detailed explanation. And I do understand why Tel Aviv University gave you the Teacher of the Year award. You're a great teacher. Anything you want to add? Something important we missed?
Yes. I have been doing this for many, many years. But to be totally honest, I don't do any work. I'm blessed with amazing, amazing students who for years are no longer my students. They are my collaborators and friends. And without a team, there's no science.
And we see this in the world. I mean, it's amazing you're acknowledging your team and your lab, but I see a lot of collaboration in the scientific community nowadays, and I think that's something to keep.
Absolutely. Absolutely.
Great. Well, Dr. Gershoni, thank you very much, and good night to you. And
thank you.
Hi from Mexico.
Ciao.